Hi everyone, welcome to this tutorial. Today I'm going to walk you through a real geochemistry analysis using Python, where we use PCA and clustering to detect spatial anomalies in multi-element geochemical data. If you are into mineral exploration, machine learning, or just want to see how real-world data gets turned into targets, you are in the right place. So we have a geochemical dataset and we want to get ready for early exploration. Here's what we need to do. We'll start with sanity check. We clean and prepare our raw geochemical data. Next, we transform the data using center log ratio, the CRL transformation, and get ready to apply principal component analysis, PCA, to reduce dimensions. Then we'll use regression and residuals to isolate unexpected geochemical behaviors, and finally cluster those anomalies with DBSCAN. Plot the maps and export the results for use in GIS, all inside a reproducible Jupyter notebook. Loading and cleaning the dataset includes handling missing values, filtering out invalid measurements, creating both log transform and raw versions of each geochemical element. And we're also going to clip extremely small values and ensure the data set is clean enough for a statistical operation. After the data set is cleaned, we apply a CLR transform. This helps with compositional nature of geochemical data. Then we run principal component analysis to reduce dimensionality. PCA captures the main geochemical trends in the datasets like regional lithology or alteration halos and separate noise and background. Here's what the PCA scatter looks like in the right panel, and we can also map these PCAs spatially to see the anomalies or trends, as you can see in the left panel here. Then is a prediction stage. Instead of just looking at raw concentration, we build a linear model to predict elements like nickel or copper using PCA components. What we are really interested in are the residuals, the difference between what the model expects and what we actually observe. So these residuals highlight local geochemical anomalies like veins, alteration zones, or mineralized pockets that do not follow the original trend. Then we scale the residuals and run DBSCAN. DBSCAN is a clustering algorithm that groups dense blobs of similar anomalies. Unlike k-means, DBSCAN doesn't need us to choose the number of clusters, and it detects outliers automatically, marked it as -1. Those outliers, there are potential mineral exploration targets. After clustering, we map the results, both clusters and outliers or anomalies, and create heat maps showing how unexpected the values are spatially. And finally, we export everything, including cluster stats, anomaly rankings, all of them into a GIS-ready CSV file. That way, we can bring our results into QGIS or ArcGIS and use them in the field. Now let's dive into the codes I prepared in Jupyter Notebook. We'll start with sanity check before start cleaning or modeling. It helps you to understand the structure and completeness of your dataset, to identify issues like missing or malformed data, and also to plan your next pre-processing steps. Now we'll go through this code block step by step. First, you must import libraries. Pandas is imported as PD, used for handling tabular data frames. NumPy is imported as NP, used for numerical operation like handling nans, infs, mathematical function, etc. Next step is load our geochemical data. Load your dataset from the file, a CSV file, into a variable called df, is a pandas data frame. Assuming this file is in the same folder as your Jupyter notebook or your script. After loading data, it's time to read data and show basic info by df.info. So this function prints a summary of data frame structure, number of rows and columns, column names, data types, is it object, float 64, information, what, and a number of non-null or non-missing entries per columns. And it's useful because it helps you to quickly identify which columns have missing data, whether data types are as expected, or if anything is misread, numbers, stores as a strings, for example. And finally, we'll generate a summary statistics by df.describe for all numeric columns in the datasets. So count here is number of non-missing values, mean is average, STD is standard deviation, and you can get minimum, maximum, quartiles, and all of the extremes. A lot of statistical information that you can use here. So this is really useful. Lets you spot extremely skewed distribution, for example, when mean is not equal to median or something like that. Columns with weird values, missing values, 0, or columns with text in the cells. Also, it helps you to investigate the basic range of each variable before going further into the data cleaning and pre-processing stage. Okay, now that we get familiar with the data set, we move forward to data pre-processing. For example, we notice that some geochemical values are presented in logarithmic form, which are not suitable for analysis, like PCA, regression, clustering. Also, we need to apply a CLR transform before PCA analysis. So doing that, I've prepared this code block, and I will go through it step by step. So in step 1, we define the columns, the metadata columns. We don't use it in PCA or modeling, but it's useful for mapping. We define geochemical columns. Geocals is a log transform, geochemical values. This is your input data. Then in step 2, fills in missing values by making a clean copy of the data set, df clean. In step 3, we fill in any nan in log columns with that column's mean, and this keeps the data usable in analysis like PCA. And now we're ready to convert log transform data back into original concentration in the next step, step 4. So we got a new column, we create a new column here, and this makes the data suitable for compositional analysis like CLR. In step 5, again, we'll ensure that there is no bad data remain. Replace infinite or 0 values with none. These 0 values can break CLR or PCA. And also we drops rows where any raw columns is missing. Clips, also here we clip very small values to avoid issues with log 0 later. Now we are ready to display output and stats in step 6. So these outputs shows what columns were kept, what columns created, shows how many rows remain after cleaning. And for this, we can use describe function to display count, mean, minimum, maximum, etc. for our newly created raw columns. And finally, in step 7, we visualize data distribution for the last time before applying CRL and PCA. You will see your histograms for the first 6 raw elements, and these plots lets you visually check normality versus skewness or outliers and an overall range of each element. Now that the pre-processing is complete and we're happy with our data set, we will apply CLR and PCA. Again, we start with adding relevant libraries. PCA from scikit-learn. This performs principal component analysis, and also we added Seaborn and Matplotlib used to create a nice scatter plots of the PCA results. In step 1, we select columns to transform. This extracts only the raw columns, creates a new data frame, the CLR input for our CLR processing, and ensure your PCA input respect the closure, the compositional data constraints. Step 2 is the CLR transformation. The function here takes each row, each sample, computes the geometric means of its elements value, replace each value with logarithmic ratio of values over geometric mean. This transforms the compositional data from the simplex to Euclidean space. And the results is stored here in CLRDF. That's your input for your PCA. And it's important because you cannot apply PCA directly to compositional data due to the constant sum constraint, which is called closure. So CLR beautifully fixed that. In step 3, we apply PCA. So for PCA, the component that we added here is 3 components. PCA tells scikit-learn to reduce the data now into 3 principal components. So fit transform applies the PCA here. PCA scores, PCA underline scores is a NumPy array with 3 new columns for each sample, and we store it in a new data frame. It's called PCA underline DF, and retain the original row indices. Now each sample is represented by 3 components, PC1, PC2, and PC3, that capture the most variation in geochemistry. So at the end of this cell, this code block, you will have this CLRDF, which is your compositional geochemistry in Euclidean space. You have the PCA-DF, the PCA data frame. Your data frame is 3 principal components and coordinates. You have the visual plot of PC1, PC2, PC3, and a handle on how much variance PCA captures. But before plotting and discussing PCAs, I would like to elaborate more on the main goals of applying PCA to our geochemical dataset. There are several benefits that it brings. For example, noise filtering that automatically de-emphasize random or minor fluctuation in data, which often appears in PC3 or PC4, etc. Redundancy removal can detect and combines correlated variable into fewer axes. For example, copper and nickel often vary together. Pattern discovery is another benefit, that the PCA reveals underlying trends, gradients, or groupings that are not obvious in raw variables. Visualization, PCA makes it easier to see complex data in 2D or 3-dimensional plots. And dimensionality reduction is one of the most important, that reduce many input variables into just a few components, like PC1, PC2, PC3, that explain most of the variation. But what does this mean in a plain language when we say most of variation? So PC1 captures the biggest patterns in how your elements vary together across samples. Let's break it down. You have thousands of samples, for example. Each sample has many geochemical elements, copper, nickel, zinc. And these elements don't vary independently. They often go up and down together due to the geological processes. So PCA looks at how all these variables covary and finds a single axis, which is called PC1, that captures the largest overall trend in the data. So analogy is that imagine you're looking at a cloud of points in 3-dimensional copper, nickel, and zinc. They may not spread equally in all directions. Instead, the cloud is elongated along a certain direction. Maybe copper and nickel increase together while zinc stays flat. PC1 here is the line or axis that best fits the long direction of this cloud, the main trend. PC2 is the next best direction perpendicular to PC1, is capturing the second biggest trend. So why variance matters? In statistics, variance is a measure of how much things differ from the average. By focusing on maximizing variance, PCA prioritizes the strongest, most important trends and can filter out noise, redundant, or less significant variations. Now I would like to dive in deeper into the concept of PCA by giving you a numerical example. It's the best way to understand what it means when PCA explains the most variance. So let's use a simple data set here. Imagine we have 10 samples and we measured 3 elements, copper, nickel, and zinc. We simulate data where copper and nickel increase together while zinc varies independently. So what's happening here is that copper and nickel increase together, very strong correlation. Zinc varies randomly, not related to copper or nickel. What PCA does here is that PCA computes variance in each direction. So PCA will align with the direction where copper and nickel vary together. PC2 will be orthogonal, capturing what's left, mostly zinc variation. PC3, if it exists, would be very small because there's a little left over. PCA then computes the percent of variation explained. So if we run PCA here, we would get something like for PC1, 0.94, or to say 94% of total variation for PC1, 5% of variation for PC2, and a tiny, maybe just 1% for PC3, just noise. This means nearly all the variation in the data can be explained by PC1, which is the copper-nickel trend. And zinc contributes only a small amount of unique variation as PC2. Now that we perfectly understand the PCA concept, we move on to print the computed results and plot them. So in step 4 in the code block, we report the variance explained. Explained underline variance underline ratio. This tells us how much variance is captured by PC1, PC2, and PC3. And NP sum here shows how much is captured cumulatively. For example, 49%, 21%, and 11% here. Actually, PC1 captures 49%, PC2 adds 21%, and PC3 adds 11%. The total is 81%. This helps you decide how many components are worth keeping for your modeling. So in the next step, we plot the PCA results to visualize each sample in PC1, PC2 space, as you can see here in this plot. And it helps you to spot natural groupings, trends, or outliers. So the alpha here is 0.6, makes overlapping points more transparent. Often different lithologies or mineralized zone will form clusters in this type of diagrams. So imagine that 3-dimensional clouds that we talked about. Now you plot PC1 versus PC2 here. You are rotating that 3-dimensional clouds to see it flattened in the direction of most variance. So you see the biggest shape in 2 dimensions. What to look for in this shape, in this plot? So in this type of plot, tight clusters or clouds often reflects similar geochemical signatures, and these might be background rock units or dominant geochemical provinces. If you see any wings, lobes, or tails, can indicate anomalies, outliers, or distinct geochemical processes like mineralization, contamination, alteration. Most of our samples here spread along PC1 vary significantly, and this means whatever pattern PC1 represents, for example, enrichment in certain metals or something else, this pattern is a dominant geochemical signal in your area. So, and also pointy, triangular, or cone or fan shaped or pattern, these kind of pattern often happens when most samples share a common base chemistry. A subset, or for example, a subset that is enriched in one or more elements. The dense cluster in the middle near 0 and 0 for PC1 and 2 may be background unaltered samples, while outliers that are away from center may be interesting anomalies for us. To better understand our geochemical datasets, I got the loadings, the eigenvectors, and column names. Then I plotted the sample scores and variable vectors. So each arrow here represents a geochemical variables, copper, nickel, zinc. The length of the arrow is equals how strongly that element influenced the PCA. The direction means which principal components it affects the most. And arrows that point in the same direction, they are positively correlated. Arrows points in the opposite directions are negatively correlated. And arrows near the origin is low contributions. So also angles between arrows can be interpreted as a correlation between elements. Small angle means strong positive correlation. For example, opposite directions is negative correlation, and 90-degree angle is no correlation. If you look at the gold here, long arrow pointing toward upper right, there's a strong positive loading on both PC1 and PC2, and this suggests gold-rich samples pull the PCA in that direction. Looking at copper, cobalt, nickel, or lead is clustered toward lower right and suggests that these elements vary together and contribute primarily to PC1. For iron, manganese, molybdenum, mercury point more toward PC2, the vertical axis, especially for manganese. These suggest these elements have a different geochemical behavior, potentially related to host rock, alteration, or environmental background. Zinc is close to the center, low influence on PC1 or PC2, weak contribution to variance compared to others. So it shows us that if gold is strong along PC1 and copper is also the same, copper might be a good proxy for gold. If iron and manganese form a separate cluster, they may represent a different lithology or mineralization style. Clustering of samples in direction of nickel means nickel-rich areas. The farther a sample is from the origin in the certain direction, the stronger its geochemical signal in that vector. And finally, the best part, which is the geospatial PCA heat map plots. I add back coordinates for plotting and add the spatial location for each sample back to the PCA results. And This is crucial for mapping, clustering, or interpreting spatial patterns in PCA space. In this case, we extracted PC1 and spatial coordinates and create a grid over the area. And the map can be your early exploration target. Then you can also overlay your PC1 heat map on a base map or satellite image using GeoPandas. And this will give you a geo-aware visual of geochemical trends that aligns with the real-world geography. So up until now, you're building a baseline geochemical model using PCA. Now we're asking, where does reality disagree with this baseline? And this disagreement highlights the local mineralization, hydrothermal alteration, or geochemical noise, and helps us for anomaly detections. So to understand these disagreements, we built linear models to predict elements like nickel, gold, copper using PCA components. And this is a simple machine learning algorithm based on a linear regression. What we're really interested in are residuals, the difference between what the model expects and what we actually observe. So in a step one, we choose the target elements. You're picking one element, nickel, that you want to predict, building a linear model using PCA. I will also show you later how to loop over other elements like copper, gold, or any other elements. In a step two, you prepare your data for regression. X is our PC1, PC2, PC3 as features. Y is our actual measured concentrations of nickel from the clean data set. So the question now is, can we explain variation in the nickel using just these three PCA components? So what we need to do here in step 3 is to train the model using a standard linear regression and find the best fit line or plane or hyperplane from the PC1, PC2 to the nickel values. And in the next step, we predict and compute the residuals in step 4. So predicted is what the model expects from nickel to be, and residual is the difference of true minus predicted. So in the step 5, we save the residuals, add a new column to our data frame, and plot our residuals on the map to see the spatial interpolation of the residuals. This is the output of this code block. Red area in your map, it means low residuals. Model over-predicted nickel. These are not the interesting anomalies. Blue areas, on the other hand, is positive residuals. Model under-predicted nickel. These are your possible mineralization targets. And it's important for us because we are detecting places where the sample doesn't follow the regional trend. Something special might have happened here. You can also do the same as above cell, but automated for multi-element predictions, for example, for gold or iron. Finally, we'll tabulate the results. Here in step 0, we define the columns you want in the final table extracted from the previous cell, and then we build the final table, get the preview, and export to CSV, which is optional. Now that we have our predictions, we can move on with clustering. Scale the residuals and run DBSCAN. DBSCAN is the clustering algorithms that groups dense blobs of similar anomalies. And clustering to the geochemical residual is quite useful to find spatial geochemical anomalies that do not follow regional scales. So DBSCAN groups nearby samples with similar unexpected behavior, similar residuals into clusters, potentially pointing to local anomalies or mineralized zone. So to do this, first we need to scale the residuals. So in step 1, we take all the residual columns, removes missing rows, standardize using standard scaler here, and this ensures equal weight for all elements during clustering. In step 2, we run DBSCAN to find dense blobs of samples that behave similarly, and the parameters that we have used for EPS is 1.2, which is how close points must be to be considered neighbors. The minimum sample is 20. You can change all of these parameters, and it will assign the cluster labels like 0, 1, 2, or label like -1 for points that do not belong to any clusters, outliers. In our case, it's most anomalous points. And then in step 3, save the cluster labels. In step 4, we plot the spatial clusters. And this is the plot for all elements, quite powerful. You're identifying multivariate anomalies, locations where geochemical signatures across copper, nickel, zinc, lead, cobalt, and other elements. It's unexpected relative to the PCA predicted trends. So anomaly could be slightly elevated nickel, moderately depleted copper, or very elevated zinc here, or any non-background combination that breaks the usual PCs or PC structures. Anyway, you can customize the plot to the element of interest, for example, only for nickel or gold, or combination of gold and copper. I can see here is the plot is only for gold. Anyway, in this plot, orange points are cluster 0. These are the dense regions where your residual patterns are similar. DBSCAN group them as one cluster. Blue points are cluster -1, and these are outliers, spatially or geochemically isolated points that do not belong to the main population, and DBSCAN label them as -1 because they have unusual residual patterns. So what this might mean in geochemical context, these blue outliers could represent mineralized zones that deviate from the background patterns of geochemical halos, veins, or structures. Now you can tabulate the results in any form you like. For example, you can have maximum residual per point for all points and clusters and export to GIS for further refinements. Here in this code block in step 1, we'll compute the maximum residual per point. You already have a residual for multiple elements, copper, nickel, lead, gold. And this line creates a new column. It's called max residual that holds the highest residual, the most anomalous for each location. And it's useful for flagging the most extreme deviations. In step 2, select columns for GIS exports, create a list of columns to export basic location, cluster information, residuals, and then we combine def clean and the PCA def into one data frame using their shared index. The results, each row has location, residuals, cluster, and maximum residuals. In step 3, we drop incomplete rows, remove rows with missing coordinates or cluster labels, ensure the export will load cleanly into GIS software. And in step 4, finally, we export your clean table to CSV, ready to import into QGIS or ArcGIS or GeoPandas. We can also investigate anomalies to understand other groups. So we want to understand the background or trend-based groupings. This code block summarizes the typical geochemical behavior of each cluster by calculating average residuals and ranking clusters by how anomalous they are. So the purpose is that to characterize and prioritize each DBSCAN derived clusters. So you'll get a table that tells you what each cluster look like in terms of mean residuals for copper, nickel, zinc, etc. And how many samples belong to each cluster. Which cluster are the most anomalous based on the highest maximum residual. And it's important to help us to identify the most promising anomalous zone and helps us to target our selections and explorations to make clustering results interpretable instead of just having label numbers. So you can now answer these questions. What's special about cluster 2, for example? Which cluster should we investigate first? And are certain anomaly dominated for nickel or copper or zinc? And this might not be important in this case, as we only have 2 clusters, but in some cases, you got several clusters that need to be statistically assessed before moving for further refinement. So what we have done here is to exclude the anomalous cluster -1, not to interfere with other group, with group 0. And the interpretation of cluster 0, the summary is here. For copper, the residual is about minus 0.012, which is slightly under predicted the mild depletion. For nickel, the residual is 0.003. Nickel is almost exactly as predicted. Zinc is minus 0.478. It shows that zinc is slightly under predicted. It's a mild depletion. And for gold is very close to the predicted. It's 0, is in grouping 0. So this cluster, the cluster 0, likely represents a background or normal geochemical pattern, the regional geochemical baseline, and these samples follow PCA trends very well. Nothing stands out strongly here in grouping 0, just small residuals. And if any other clusters like cluster 1, 2, cluster -1 have much higher maximum residual and fewer points, those are your real anomalies. You can have the same table for the anomalous cluster -1, here as below. Here the number of points for group 0 is about 26,000 versus point count for group -1, which is about 3,000. The maximum residual for group 0 is 36, and the maximum residual for group -1 is 187. So we can say that the group -1 is the real anomaly, just from looking at the statistics in the table. So what have we done? We started with a messy geochemical data and ended with the interpretable ranked geochemical anomalies ready for mapping and fieldwork. This workflow is transparent, reproducible, and totally customizable for other data sets or projects. And if you found this helpful, give it a like, leave a comment with questions, and don't forget to subscribe for more geoscience and data tutorials. Thank you very much.